The urinary system. So the urinary system is the major excretory system of the body. The urinary system consists of two kidneys, two ureters, the urinary bladder, and the urethra. So each kidney waste products are carried by a ureter to a single urinary bladder. So the waste is emptied from the urinary bladder by the urethra. So the kidneys each filter a large volume of blood. So waste from the blood are collected to form urine. And the urine consists of excess water, excess ions, metabolic waste, and toxic substances. So the kidney can suffer extensive damage and still maintain homeostasis. As long as about 1/3 of one kidney remains functional, survival is possible. However, if the functional ability of the kidney fails completely, death will result unless the person receives medical treatment. So the major function of urinary system is excretion, which controls the composition of volume of body fluids. The kidneys also have many other important metabolic activities. So the kidney function includes number 1 is excretion, number 2 is regulation of blood volume and pressure, number 3 is regulation of blood solute and concentration, then number 4, the regulation of extracellular fluid pH, then number 5, regulation of red blood cell synthesis, and the last one is regulation of vitamin D synthesis. And external anatomy of the kidney. So the kidney are behind the peritoneum or the retroperitoneal and are located each side of the vertebral column. So they are the bean-shaped organs, each about size of a tightly clenched fist. Internal anatomy and histology of the kidney. So to fully appreciate the function of the kidney, we must understand their ultrastructure. So the two kidneys are organized into two major regions. Number 1 is an outer cortex and an inner medulla. The cortex is the location for the blood filtering structures of the kidney, and the medulla is composed of many cone-shaped structures called renal pyramids, whose bases project into the cortex. Urine is formed in the cortex and medulla, and enters the calyces from the tips of the renal pyramids. From the calyces, urine flows into the renal pelvis and then exits the pelvis through the ureter. Urine flows through the ureter. Urine flows through the ureter to the bladder, where it is stored. When the urine is released from the bladder, it exits through the urethra into the external environment. Structure of a nephron. The nephron is the histological and functional unit of the kidney. It consists of specialized structures, including small tubes that are called tubules. There are approximately 1.3 million nephrons distributed through the cortex and medulla of each kidney. The four regions of a nephron are a renal corpuscle, a proximal convoluted tubule, a loop of Henle, and a distal convoluted tubule. There are two types of nephrons in the kidney: the juxtamedullary nephrons and the cortical nephrons. So the juxtamedullary nephrons have renal corpuscle that are found deep in the cortex near the medulla. And the cortical nephrons have renal corpuscle that are distributed through the cortex. The renal corpuscle. The filtration portion of the nephron is housed in the renal corpuscle. The renal corpuscle consists of one glomerulus and the Bowman capsule. The glomerulus is a network of capillaries twisted around each other. The Bowman capsule is an intended double-walled chamber surrounding the glomerulus. Together, the structure and the corpuscle make up the filtration membrane. The filtration membrane performs the first major step in urine production. Urine production begins when the filtration membrane filters the blood. The filtered fluid, called filtrate, then enters the lumen or space inside the Bowman capsule. An important regulatory structure called the juxtaglomerular apparatus is located next to the glomerulus. Secretion of the enzyme renin by the Juxtaglomerular apparatus plays an important role in the regulation of filtrate formation and blood pressure. The renal tubule. So the renal tubule collects the products that are filtered out from the blood. Arteries and veins. A system of blood vessels allow the exchange of materials that occurs in the kidney. The renal arteries branch off the abdominal aorta and enter the kidneys. As the renal artery extends deep into the kidney, it branches into smaller and smaller blood vessels. The following is a list of blood vessels in order, starting with the renal artery and progressing to the blood vessels that surround each nephron. Number 1. The interlobar arteries pass between the renal pyramids. Number 2. The articulate arteries branch from the interlobar arteries. They arch between the cortex and the medulla. Number 3. Interlobular arteries branch off the articulate arteries and project into the cortex. Number 4. The afferent arterioles arise from branches of the interlobar arteries. The afferent arterioles carries blood to the glomerular capillaries. Number 5. Efferent arterioles carry blood from the glomerular capillaries. And number 6. The peritubular capillaries branch from the efferent arterioles. Blood from the peritubular capillaries, including the vasa recta, will return to the general circulation through the veins of the kidneys.
The primary function of the kidney is regulation of body fluid composition. The kidney is the organ that sorts the substances from the blood for either removal in the urine or return to the blood. Substances that are waste products, toxins, and exist materials are permanently removed from the body, whereas other substances need to be conserved to maintain homeostasis. The structural components that perform this sorting are the nephrons, the functional unit of the kidney responsible for filtering blood, removing waste products, and regulating electrolytes balance to produce urine.
Urine formation involves three major processes. First is the filtration. Filtration occurs within the renal corpuscle. Second is the reabsorption. Reabsorption happens primarily in the proximal convoluted tubule and the lobe of Henle, although there is some reabsorption in the distal convoluted tubule. The last process is the secretion. Secretion occurs mostly in the distal convoluted tubule, but may also occur in the proximal convoluted tubule.
Filtration. Blood pressure in the glomerular capillaries forces fluid and small molecules out of the blood to create filtrate. Filtration is non-selective and separates based only on size or charge of molecules. Filtration is comparable to emptying your junk drawer of everything except large, permanent items. Filtration does not remove everything in the blood. Filtration removes only those substances small enough to fit through the filtration membrane.
In all the nephrons, both of the kidneys, about 180 liters of filtrate are produced each day, but only about 1% or less of the filtrate becomes urine because most of the filtrate is reabsorbed. Recall that the renal corpuscle and the renal cortex contain the filtration structures, each of which is called a filtration membrane. The membrane separates material on the basis of size and charge of the blood components. Thus, the filtration membrane allows water and small molecules to leave the blood while preventing blood cells and most protein from leaving the blood. For the filtration membrane, three forces or pressures determine the amount of filtrate formed. Combination of these three pressures is called filtration pressure.
Glomerular capillary pressure. The glomerular capillary pressure is essentially the blood pressure inside the glomerular capillaries. It is an outward pressure from blood pressing on the fenestrated capillary walls. The glomerular capillary pressure forces fluid and solutes out of the blood into the Bowman capsule. This pressure is higher than that in other capillaries of the body due to the smaller diameter of the efferent arteriole compared to that of the efferent anterior and glomerular capillaries. Capillary absorption is the process of water and solutes from the filtrate into the blood. Proper tubular absorption is critical in preventing the body from becoming overly dehydrated and deficient in important materials. Nearly all 99% of the water and solutes are rapidly returned to the blood via the renal tubules. The filtrate leaves the lumen of the Bowman capsule and flows first through the proximal convoluted tubule into the loop of Henle and the distal convoluted tubule, and then finally into the collecting ducts. The proximal convoluted tubule is the site of the majority of reabsorption. The mechanisms underlying reabsorption can be better understood by considering the cells found there. The cells of the proximal convoluted tubule have numerous microvilli, which dramatically increase the surface area available for reabsorption. Reabsorption of most solutes is linked to the diffusion of sodium into the cells of the proximal convoluted tubule. There is a step concentration gradient for sodium from the filtrate into the cytoplasms of the cells of the proximal convoluted tubule. This concentration gradient is established by active transport of sodium out of the cells of the proximal convoluted tubule. This movement of sodium into these cells is responsible for the transport of many other solutes from the lumen of the proximal convoluted tubule into the cytoplasms of the tubule cells. Carrier molecules that transport amino acids, glucose, and other solutes are located within the proximal convoluted tubule cells. Each of these carrier molecules binds specifically to one of those substances to be transported and to sodium. The concentration gradient for sodium provides the energy that moves both the sodium and the other molecules or ions from the lumen into the tubule cell. Once the molecules are inside the cell, they enter the interstitial fluid by facilitated diffusion or contrasport. The number of the carrier proteins limits the rate of which a substance can be transported. Solutes are transported out of the lumen through the proximal convoluted tubule cells and into the interstitial fluid. Water follows by osmosis. By the time the filtrate has reached the end of the proximal convoluted tubule, its volume has been reduced by approximately 65%. However, because the proximal convoluted tubule is permeable to water, the concentration of the filtrate there remains about the same as that of the interstitial fluid. Earlier, we described the loop of Henle and its two limbs. The two limbs differ in the type of epithelial tissue present in each other. This difference in cell type is linked to the permeability of each limb to water and solutes. Reabsorption in the descending limb of the loop of Henle. The epithelial tissue in the majority of the descending limb, in particular the 10 segment, is simple squamous epithelial tissue. Remember from chapter 4 that simple squamous cells are highly permeable to water, which means the descending limb is highly permeable to water. Water moves by osmosis out of the descending limb, while some solutes move by diffusion into the descending limb. By the time the filtrate has reached the end of the thin segment, the volume of the filtrate has been reduced by another 15% and its concentration has significantly increased to 1,200 liters. Reabsorption in the ascending limb of the loop of Henle. As the loop of Henle makes its hairpin turn into ascending limb, the simple squamous epithelium persists, but it has become impermeable to water. However, it is still permeable to solutes, which exit the ascending limb, thereby again reducing the concentration of the filtrate. As the ascending limb continues, it is now called the thick segment. The thick segment of the ascending limb is impermeable to both water and solutes. Instead, the cells of the thick segment house multiple types of transport proteins, including ATP-powered pumps and carrier molecules. These carrier molecules remove a significant portion of the solutes from the filtrate, which then enters the interstitial fluid. It is this active transport of solutes that contributes to the kidney's ability to conserve water. Co-transport is responsible for moving potassium and chlorine with sodium across the membrane of the ascending limb of the loop of Henle. Once inside the cells of the ascending limb, chlorine and potassium exit the cells of the ascending limb via facilitated diffusion. As we follow the filtrate through the loop of Henle, we see that it becomes very concentrated toward the hairpin turn off the loop of Henle, but the concentration of the filtrate is reduced to about 100 liter by the time the fluid reaches the distal convoluted tubule. Distal convoluted tubule and collecting duct. Some solutes, like calcium and hydrogen, are not reabsorbed until further along the renal tubule in the distal convoluted tubule or collecting duct. Their absorption of these solutes is generally under hormonal control and depends on the current condition of the body. The distal convoluted tubule and the collecting duct are not always permeable to water. However, hormone regulation can change permeability to water. Tubular secretion. Certain tubule cells transport additional solutes from the blood into the filtrate. Some of these solutes may not have been filtered by the filtration membrane, as if these are some throwaway items that had been left behind in your junk drawer. As with tubular absorption, tubular secretion can be either active or passive. Urine concentration mechanism. The kidneys are able to produce by dilute or very concentrated urine while maintaining the extracellular fluid concentration very close to 300 liter. But how does the kidney move such a large volume of fluid from the blood into the filtrate and then back into the blood? The kidney's ability to control the volume and concentration of the urine depends on three factors. First is the counter-current mechanisms. Second is a medullary concentration gradient. And the last one is the hormonal mechanisms. The counter-current mechanisms. The kidneys utilize a counter-current mechanisms. A counter-current mechanism is one where fluid in separate structures flow in opposite directions relative to each other. As the fluids pass by each other, materials can be exchanged between the fluids. Medullary concentration gradient. The interstitial fluid in the medulla of the kidney has a very high solute concentration compared with that of the cortex. This is called the medullary concentration gradient. The high solute concentration of the interstitial fluid develops from the action of the countercurrent mechanisms and the recycling of the protein breakdown product, urea. To create and maintain the medullary concentration gradient, the following processes occur. First is the countercurrent mechanism. As filtrate travels through the descending limb of the loop of Henle, water moves across its simple squamous epithelium by osmosis toward the higher solute concentration in the interstitial fluid. Second is the urea recycling. Urea is responsible for a substantial part of the high solute concentration in the kidney medulla. Urea is recycled from the interstitial fluid through the renal tubules and back into the interstitial fluid.
We have a fascinating topic to delve into: the regulation of urine concentration and volume. Now, this might sound a bit technical, but trust me, it's a topic that touches our everyday life. Our bodies are like well-oiled machines, constantly working to maintain a balance of fluids and electrolytes. One of the ways it does this is through the production and regulation of urine. So, whether you're a medical professional, a student, or just someone curious about how your body works, this talk show is for you. Stay tuned as we dive into the fascinating world of urine regulation. Regulation of urine concentration and volume. Have you ever wondered how your body maintains the right balance of water and solutes? Let's dive into the fascinating world of urine concentration and blood pressure regulation. The kidneys play a crucial role in regulating the concentration of our body fluids. They have the remarkable ability to adjust the amount of water reabsorbed, resulting in urine that can be either concentrated or dilute. Inside the kidneys, tiny structures called nephrons are responsible for this important task. As fluid passes through the nephron, the kidneys carefully monitor its concentration. When our body fluid concentration rises, the kidneys step in to conserve water. They reabsorb more water from the fluid passing through the nephron, resulting in a smaller volume of concentrated urine. This helps eliminate excess solutes and brings our body fluid concentration back to normal. Conversely, when our body fluid concentration decreases, the kidneys adjust their water reabsorption. They reabsorb less water, allowing a larger volume of dilute urine to be produced. This helps conserve solutes and increase the body fluid concentration. But wait, there's more. The kidneys also play a vital role in regulating our blood pressure. You see, urine production affects our blood volume, and changes in blood volume can affect blood pressure. When our blood volume is above normal, the kidneys produce a large volume of urine. This helps lower blood volume and subsequently reduces blood pressure. That's why diuretics, which increase urine production, are commonly used to lower high blood pressure. On the other hand, when our blood volume is below normal, the kidneys adjust their urine production accordingly. They produce a smaller volume of urine, helping increase blood volume and raise blood pressure. So, you see, our kidneys are true superheroes, working tirelessly to maintain the delicate balance of water and solutes in our bodies. They ensure that our urine concentration is just right and our blood pressure stays within a healthy range. Next time you take a sip of water, remember to thank your incredible kidneys for their incredible work in keeping you healthy and balanced. Hormonal mechanisms. Today, we'll explore the three major hormonal mechanisms that work together to maintain the delicate balance in our bodies. These mechanisms are the renin-angiotensin-aldosterone mechanism, the antidiuretic hormone or ADH mechanism, and the atrial natriuretic hormone mechanism. Let's dive in and understand how they keep our urine concentration and volume just right. Our first mechanism is the renin-angiotensin-aldosterone mechanism. This mechanism is more sensitive to changes in blood pressure. When blood pressure drops, Specialized cells in the kidneys release an enzyme called renin. Renin acts on a protein called angiotensinogen, converting it into angiotensin I. This inactive form then travels to the lungs, where another enzyme converts it into angiotensin II. Angiotensin II is a powerful hormone that stimulates the release of aldosterone from the adrenal glands. Aldosterone acts on the kidneys, promoting the reabsorption of sodium and water, while increasing the excretion of potassium. This leads to increased blood volume and subsequently raises blood pressure. Now, let's move on to the ADH mechanism. This mechanism is more sensitive to changes in blood concentration. When blood becomes too concentrated, specialized cells in the hypothalamus release antidiuretic hormone or ADH. ADH travels to the kidneys and acts on the tubules, increasing their permeability to water. This allows more water to be reabsorbed back into the bloodstream, reducing urine volume and concentrating it. Last but not least, we have the atrial natriuretic hormone or ANH mechanism. This mechanism responds to changes in blood pressure, particularly when it becomes too high. When the heart's atria detect high blood pressure, they release ANH. This hormone acts on blood vessels to dilate them, reducing blood pressure. It also acts on the kidneys, inhibiting the reabsorption of sodium and water, promoting their excretion in urine. This helps lower blood volume and pressure. These three hormonal mechanisms, the renin-angiotensin-aldosterone mechanism, the ADH mechanism, and the ANH mechanism, work in harmony to maintain the balance of urine concentration and volume in our bodies. The renin-angiotensin-aldosterone mechanism. This mechanism plays a crucial role in regulating blood pressure and volume in our bodies. Let's explore how it works. Our story begins in the kidneys, where specialized cells called juxtaglomerular cells monitor blood pressure. When blood pressure drops, these cells spring into action. The juxtaglomerular cells release an enzyme called renin into the bloodstream. This occurs in response to reduced stretch of the arteriole, which supplies blood to the kidneys. Renin then acts on a plasma protein called angiotensinogen, which is produced by the liver. It converts angiotensinogen into angiotensin. But our story doesn't end there. Angiotensin needs a little more transformation. It travels to the lungs, where an enzyme called angiotensin converting enzyme, or ACA, converts it into its active form, angiotensin. Angiotensin is a powerful hormone with multiple effects. First, it acts on blood vessels, causing them to constrict. This vasoconstriction helps increase blood pressure. Angiotensin II also stimulates thirst, making us feel thirsty and encouraging us to drink more fluids. It also increases salt appetite, making us crave salty foods. But that's not all. Angiotensin stimulates the adrenal cortex, a part of our adrenal glands, to secrete a hormone called aldosterone. Aldosterone's main job is to promote the reabsorption of sodium and chloride ions in the distal convoluted tubules and collating ducts of the kidneys. This reabsorption leads to the reabsorption of water. increasing blood volume and returning blood pressure back to the normal range. And there you have it. The renin-angiotensin-aldosterone mechanism helps regulate blood pressure and volume by increasing blood pressure, stimulating thirst and salt appetite, and promoting the reabsorption of sodium and water. It's a remarkable system that keeps our bodies in balance. The antidiuretic hormone mechanism. Our story begins when there is an increase in solute concentration in the blood or interstitial fluid. Events like changes in solute concentration can trigger the release of ADH. ADH, also known as vasopressin, is released by the posterior pituitary gland in response to these changes in solute concentration. ADH travels through the bloodstream and acts on the kidneys. It increases the permeability of the distal involuted tubules and collecting ducts to water. Increasing water permeability, more water is reabsorbed from the filtrate in the kidneys. This leads to the production of a small volume of concentrated urine. Conservation of water in the kidneys helps prevent excessive water loss, especially when solute concentration is high. The result is a reduction in urine volume. On the other hand, when blood solute concentration is low, ADH secretion is inhibited. This leads to a decrease in water reabsorption in the kidneys and an increase in urine volume. But that's not all. ADH secretion is also regulated by changes in blood pressure. Arterial natriuretic hormone. Our story begins when there is an increase in blood pressure. This triggers the release of the atrial natriuretic hormone or ANH from specialized cardiac muscle cells in the right atrium of the heart. Blood pressure in the right atrium increases above normal. ANH is secreted into the bloodstream. ANH then acts on the kidney, specifically on the nephron, the functional units of the kidneys. One of the main effects of ANH is to decrease the reabsorption of sodium in the nephrons. As a result, sodium remains in the tubules of the nephron. With sodium remaining in the tubules, water moves by osmosis from the surrounding tissues into the tubules, following the concentration gradient of sodium. This increase in water moving into the tubules leads to an increase in urine volume. Increased urine volume results in a reduction in blood volume and subsequently lowers blood pressure. Bladder cancer is a relatively common type of cancer, but the good news is that the 5-year survival rate is high if detected and treated early. Intriguing treatment method for early-stage bladder cancer involves introducing a fluid containing live tuberculosis-causing bacteria into the bladder. Surprisingly, this treatment has shown effectiveness in some cases. The live bacteria stimulate the immune system, helping it recognize and destroy cancer cells. Important to note that cigarette smoking is a major risk factor for bladder cancer, even years after quitting. The first sign of bladder cancer is often blood in the urine. To confirm the diagnosis, a procedure called cystoscopy is usually performed. In cystoscopy, a catheter is inserted into the bladder to collect cells for examination. This helps determine the presence and extent of cancerous cells. Treatment for bladder cancer depends on the depth of tumor penetration into the bladder wall. Early stages, cancerous cells can be scraped off the bladder wall. This procedure aims to remove the abnormal cells and prevent further growth. Cases, a form of immunotherapy called intravesicular delivery of bacillus calmette Jeann or BCG is used. It is a weakened strain of the bacterium that causes tuberculosis in cattle, but it can also infect humans. The treatment involves infecting the cancer cells with BCG bacteria. The presence of BCG bacteria stimulates the immune system to recognize and destroy the infected cancer cells. Then typically lasts for about 6 weeks. The use of bacteria in bladder cancer treatment showcases how knowledge of body systems can be applied in unexpected ways. By harnessing the power of the immune system, we can target and destroy cancer cells, offering hope for patients with early-stage bladder cancer. The role of maturation reflex by higher brain centers. Maturation or urination can be voluntarily initiated through an increase in action potentials from the cerebrum. This facilitates the maturation reflex and relaxes the external urethral sphincter of the abdominal muscles, also plays a role in enhancing the maturation reflex. Pressure on the bladder wall, aiding in the expulsion of urine. The need to urinate occurs due to the stretch of the bladder. When the bladder becomes sufficiently stretched, nerve fibers send signals to the brain, signals to urinate. Irritation of the bladder or urethra can trigger the urge to urinate. Even if the bladder is not full, this irritation can arise from various factors such as inflammation or infection. Urination reflex is an automatic process that causes a decrease in action potentials sent to the external urethral sphincter. Relaxation. Higher brain centers have the ability to inhibit or stimulate the maturation reflex. They can decrease the intensity of the reflex that stimulates bladder contractions and stimulate nerves that keep the external urethral sphincter contracted. Voluntarily inhibit maturation develops around the age of 2 to 3 years. This allows individuals to consciously or control the urge to urinate. Shift our focus to kidney stone prevention. Kidney stones or renal calculi can cause severe pain in the abdominal and inguinal regions. Contractions of the ureter, the narrow tube connecting the kidneys to the bladder, push kidney stones through this passage. This can lead to inflammation and pain. Stones are composed mainly of calcium oxalate. Preventing their formation is crucial for maintaining urinary health. Get amount of fluids, at least 2 liters per day, can help prevent the formation of kidney stones. Dilutes the urine, reducing calcium salts, precipitating and forming stones. These stones withdraw the common treatment. This non-invasive procedure uses ultrasound waves to break the stones into small particles that can easily pass through the. Let's look at the two major compartments, the intracellular fluid compartment and the extracellular fluid compartment. Compartment refers to the fluid inside our cells. It makes about 70% of our body's water and roughly 30% of our total body weight. This fluid has a similar composition in all cells, which helps maintain balance and proper functioning. The cellular fluid compartment is where things get interesting. It accounts for around 30% of our body's water and about 20% of our total body weight. This compartment includes the fluid outside our cells and is further divided into different subcompartments. Is the interstitial fluid, which is the fluid found between our cells. It surrounds our cells and provides them with nutrients and removes waste products. Is the plasma, which is the fluid component of our blood vessels. Plasma carries important substances like oxygen, nutrients, and hormones throughout our body. Compartments within the extracellular fluid, such as lymph, lymphatic vessels, cerebrospinal fluid in our brain and spinal cord, and synovial fluid in our synovial joints. However, the interstitial fluid and plasma are the main components of the extracellular fluid. And ions in these compartments is crucial for maintaining balance and proper functioning in our body. It helps regulate things like hydration, electrolyte levels, and overall homeostasis. So, the next time you take a sip of water, remember that it's not just quenching your thirst, but also playing a vital role in maintaining the balance of fluids and ions in your body's different compartments. Look at the composition of the fluid inside and outside our cells. Our body's fluids can be divided into two main compartments, the intracellular fluid and the extracellular fluid. Refers to the fluid that's inside our cells. Interestingly, the composition of this fluid is quite similar among different cells in our body. It contains a relatively high concentration of certain ions such as potassium, magnesium, phosphate, in various cellular processes. In contrast, the concentration of other ions like sodium, calcium, chloride, and bicarbonate are fluid compared to the extracellular fluid. These ions are more abundant in the fluid outside the cells. Hydration is essential for maintaining proper cellular function and communication. Is the protein concentration. The intracellular fluid has a higher concentration of proteins compared to the extracellular fluid. Protein cellular functions such as enzyme activity structural support, and transportation of molecules. Focus to the extracellular fluid. This fluid is found outside our cells but within our body. It maintains a relatively consistent composition throughout different areas of our body. The concentration remains similar in the extracellular fluid, regardless of the specific location. In the extracellular fluid, our body ensures that cells can communicate and function properly. Our fluid has a similar composition among different cells with higher concentrations of specific ions and proteins. On the other hand, the extracellular fluid maintains a consistent composition throughout the body with similar concentrations of ions and proteins in different areas. The position of these fluid compartments helps us appreciate the intricate balance required for our cells to function optimally and maintain overall bodily health. Between our body. The cell membranes that separate these compartments are selectively permeable, meaning they allow water to pass through but restrict the movement of such as ions and molecules. By two main factors: hydrostatic pressure and osmotic differences. Is the force exerted by fluids like blood against the walls of blood vessels. Osmotic differences refer to the concentration of solutes such as ions and proteins. Indifferent exchange of fluids between capillaries and the interstitial space. At the arterial end of the capillary, where blood pressure is higher, water moves from the capillary into the interstitial space. This movement is driven by the hydrostatic pressure. At the venous end of the capillary, where osmotic pressure is higher inside the capillary, water returns to the capillary. The osmotic pressure. The total osmotic pressure in each compartment is generally this allows for continuous exchange of water and electrolytes between the compartments. One compartment changes significantly, the compartment with a higher solute concentration. This osmosis, where water diffuses towards a solution with a higher solute concentration. Osmosis plays a crucial role in maintaining fluid compartments. It helps ensure that the right amount of water and electrolytes are present in each compartment. Cellular function and overall body homeostasis. During an inflammatory response or dehydration, the permeability of the capillary wall may change, and water between the plasma and interstitial fluid, resulting in swelling or edema. Similarly, water may move into the plasma to compensate for the lack of fluid. In summary, the regulation of fluid balance and osmosis is vital for maintaining homeostasis in our body. The selective permeability of cell membranes, along with hydrostatic pressure and osmotic depth chain for the exchange of fluids between compartments, ensuring that our cells receive the necessary nutrients and maintain their proper function.
Regulation of extracellular fluid composition. Homeostasis requires that intake of substances equals their elimination. Needed water and ions enter the body by indigestion. Exist water and ions exist the body by excretion. The amount of water and ions entering the level of the body can vary over the short term. For example, the greater quantities of water and ions are lost in the form of perspiration on warm days than on cold days, and varying amount of water and ions may be lost in the form of phases. However, over a long period, the total amount of the water and ions in the body does not change unless the individual is growing, gaining weight, or losing weight, regulating the participation of the several organ system. But the kidneys are the most important, with the skin, liver, and digestive tract playing supported roles. Two mechanism helps regulate the level of the ions in the extracellular fluids. First things is thirst regulation and ion concentration regulation. Thirst regulation, water intake is controlled by neurons and the hypothalamus, collectively called the thirst center. Thirst is the sensation that induces an urge to drink water or liquid. An increase in thirst is usually associated with a reduction in the water content of the body. For example, the mucosa of the mouth becomes dry. The thirst center is activated. Thirst, one of the important means the regulating extracellular fluid volume and concentration. Ion concentration regulation, if the water content or concentration of ion in the extracellular fluid deviates from its normal range, cells cannot control the movement of substances across their cell's membrane or the composition of their extracellular fluid. The consequence is abnormal cell functions or even cell death, keeping the extracellular fluid composition within a normal range and therefore required to sustain life. Regulating the concentration of passively charged ions such as the Na, Ka, and Ca in the body fluids is particularly important. Action potential muscle contraction and normal cell membrane permeability depend on the maintenance of narrow range of concentration for these ions. Important mechanism control the concentration of these ions in the body. Negatively charged ions are attracted to the passively charged ion. When the positive charged ion are transported, the negative charged ion move with them. Sodium ion. Sodium ion or Na are the dominant ion in the extracellular fluid. Osmotic pressure is the force that causes the neat movement of the water molecule toward an area with a higher solute concentration, solution with a higher solute concentration that have a greater osmotic pressure than solution with a lower solute concentration. About pressure, about 90 to 95% of the osmotic pressure of the extracellular fluids result from Na or sodium ions and form the negative ions associated with them. The recommended intake of sodium is 2.4 grams per day because of association with high blood pressure in some people. Most people in the United States consume 2 to 3 times by recommended amount of sodium. The kidney provide a major route by which the excess Na exit. Sodium ions are also excreted from the body in perspiration or sweat. Yes. Normally, only a small quantity of Na is lost each day in the form of sweat. Blood pressure is increased. The wall of the heart, particularly in the right atrium, are stretched. The increased stretch causes a specialized cell in the right atrium to create the hormone ANH, specialized cells in the right atrium to create ANH. ANH acts on the kidney to increase the urine production by inhibiting the reabsorption of sodium. It also inhibited the effect of ADH on the distal convoluted tubules, and collect. Potassium ions. Electrically excitable tissues such as muscles and nerves are highly sensitive to high change in the extracellular key concentration. The extracellular concentration of key must maintain within the narrow range for this tissue to function normally. Aldosterone plays a major role regulating the concentration of key and the extracellular fluid, dehydration, circulatory system shock resulting from plasma loss, and tissue damage due to the injuries such as serves burn, all cause of the extracellular key concentration to the increase above normal. In response, aldosterone secretion from the adrenal cortex increase and cause key secretion to increase. A low K concentration is the cellular fluid slow aldosterone creation from the adrenal cortex without aldosterone and kidney secreted. Next is calcium ion. The extracellular creation of calcium, like that other ions, is maintained within a narrow range. Changes in the extracellular concentration of calcium have dramatic effects of electrical properties of excitable tissue. For example, decreased extracellular calcium concentration makes cell membranes more permeable to sodium, thus making them more electrically excitable. Decreased extracellular concentration of calcium causes spontaneous action potential in nerves and muscle cells, regulating a hyper excitability and muscle tetany. Increased extracellular calcium concentration makes cell membrane less permeable to sodium, thus making them less electrically excitable. Increased extracellular concentration of calcium inhibit an action potential in nerve and muscles, regulating in a reduced excitability and either muscles weaknesses or paralysis. There are three major homeostases that regulate blood levels of calcium. First is parathyroid hormone, second is vitamin D, third is calcium. Regulation of acid-basis balance. The concentration of H or hydrogen in the body fluids is reported as the pH. The body fluid pH is maintained between 7.35 and 7.45. Any deviation from the range and life-threatening. The mechanism that regulates body fluids are critical for survival. The pH of body fluid is controlled by three factors. First is buffer system, second is respiratory system, and the third is a kidney. When the pH of the body fluid is not properly maintained, the result is acidosis or alkalosis. Regulation of acid-base balance by buffer system. Buffer are the chemical that resist a change in the pH of the solution within either acid or bases are added to the solution. The buffer is the body fluids contain salts and either weak acids or weak bases that bind to hydrogen. When hydrogen increase, those fluids or release hydrogen. When hydrogen decrease in those fluids. Buffer tends to keep the hydrogen concentration and thus the pH within a narrow range value because of the characteristic. The three major buffers in the body fluids are the protein buffer system, the PO or the phosphate in the body fluids bind reversibly to hydrogen. The binding of excess hydrogen to protein and phosphate prevents decrease in pH. Conversely, when the hydrogen concentration decreases, protein and phosphate release hydrogen, which prevents balance by the respiratory system. The respiratory system responds rapidly to change the pH and help bring the pH of body fluids back toward normal. Neurons in the respiratory center of the brain are sensitive to CO levels and the acidic pH. High CO levels and low pH cause an elevated rate of depth of respiration. As a result, CO is eliminated from the body through the lungs at a greater rate, and the concentration of CO in the body fluids decreases. As CO levels decline, the concerned transition of H also declines. The pH therefore rises back toward its normal range. Under stressful conditions, some people hyperventilate, what effect will rapid rate of ventilation have on the pH of the body fluids. And in addition, why might a person who hyperventilating benefits from the breathing into the paper bag? Low blood CO levels and increased body fluids pH reduces rate and depth of respiration. CO is eliminated from the body is reduced carbon dioxide when accumulates in the body fluids because it is continually produced as a byproduct of metabolism. As CO accumulates in a body fluid, so does hydrogen, resulting in the decreases pH.